This video abstract is AI generated. How does enzyme 1 pay the entropic cost required to become catalytically active? Enzyme 1 is the gateway of the bacterial phosphotransferase system. It uses phosphoenolpyruvate, or PEP, to initiate phosphoryl transfer through histidine 189 and launch sugar uptake and signaling in bacteria. But enzyme 1 is not active as a simple rigid structure. Its activity depends on several linked conformational equilibria. The protein can exist as a flexible monomer or as a dimer. Within the dimer, the EIC domain samples compact and expanded states, and full activity requires closure into the catalytically competent state. In this work, we asked how these structural transitions are coupled to changes in conformational entropy. To answer that question, we used a high-pressure strategy. Pressure shifts equilibria toward states with smaller partial volume. An engineered enzyme 1 variant, called 3-MEI, behaves normally at ambient pressure but dissociates into a monomer at high pressure, allowing us to directly populate and study the monomeric state. We then followed this transition with high-pressure NMR and high-pressure SAX. Solution NMR spectra showed a clear pressure-dependent shift from dimeric signals to monomeric signals. The transition was centered at about 1.5 kilobar. SAX measurements independently confirmed the same effect, showing a reduction in overall size consistent with monomer formation. These data establish that 3-MEI becomes predominantly monomeric above roughly 1.5 kilobar. Next, we asked what this means thermodynamically. Comparison with the isolated EIC domain revealed that full-length enzyme 1 forms a less stable dimer than EIC alone. This means that adding the EIN domains introduces an additional entropic penalty during dimerization. In other words, the full-length protein must pay an extra conformational cost to organize its domains. To understand that cost structurally, we combined coarse-grained molecular dynamics with RDC-guided ensemble refinement. The resulting monomeric ensemble showed that the individual domains remain folded. The EIC domain does not unfold. Instead, the major effect of monomerization is increased interdomain flexibility, especially a broader range of orientations of the EIN alpha domain relative to EIC. This leads to the final model. Enzyme 1 activation is a progressive entropy quenching pathway. Dimerization reduces interdomain flexibility and EIC disorder. PEP binding further orders the EIC domain, and final domain closure produces the catalytically competent state. Together, these results show that enzyme 1 activation is driven by stepwise restriction of conformational freedom, linking dimerization, ligand binding, and catalytic competence through progressive loss of conformational entropy. To know more, read our work online.